Good afternoon, everyone. We sorted out the lights, so the show is about to start. Before we start, I would like to introduce the speaker today. The speaker is David Levin, and he is a professor in the Department of Biosystems Engineering in the Faculty of Agricultural and Food Sciences. His research focuses on biotechnologies for sustainability, such as biofuels, biodegradable polymers, and microbial production of antitoxicants. His research is multidisciplinary. It integrates microbiology, biotechnology, and genome sciences with bioprocess and biosystem engineering. Dr. Levin has been the academic lead and co-lead on several Genome Canada funded projects. He also led the hydrogen production and purification team of the NSERC funded Hydrogen Canada and was the Prairie Platform leader within Biofuel Net, a pan-Canadian research network funded by the Network Centers of Excellence. So with that, please welcome David Levin.
Great. Thank you very much, Annika. It's a pleasure for me to be here. I grew up in Winnipeg, but then I went away from my university, and my first real job was at the University of Victoria in British Columbia, and I was there for 15 years. But I've always been a prairie kid at heart, so eventually I came back, and I'm happy to be back in Winnipeg. So the flyer that was sent out just said biotechnology for sustainability, but I added bioengineering because I'm in the Department of Biosystems Engineering, and I guess I should mention that. So really what we do is biotechnology and bioengineering for sustainability, and as the intro said, we kind of integration of microbiology and genome sciences with biosystems, bioprocess engineering. So, we all know that we're facing, that humanity, our planet, our civilization is facing one of the several great looming crises. The world population is expanding. By 2030, there'll be 7 billion people. By 2050, 9 billion people. That's going to increase, and in the developing world, there's an increasing demand for a higher standard of living, which means more consumption of energy and resources. We're facing climate change. The Earth is heating up disproportionately. Some places are getting cooler. Apparently, we're in for a very cold and long winter in Winnipeg, in Winterpeg. But this past year, 2019, was the warmest globally on record ever, beating 2018, which was previously the warmest. So things are changing. And there's some major serious issues that we have to face in terms of desertification and changes in agriculture because of changes in precipitation and temperature. And we're drowning in plastic. Over 380 million metric tons of plastic produced as of 2017. I think by now, 2019 is well over 400 million. And 57 or 47 to 50% of that winds up in landfills. A large proportion of that is low-density polyethylene, which is the most abundant single-use plastic that's produced. And a lot of it winds up in the environment. You may go to the landfill, but then blown away into fields, and ultimately into the ocean. 60% of all plastic bags are made from low-density polyethylene. 20% of all the plastic waste in landfills is low-density polyethylene. And we have a major issue with nanoparticles of polyethylene and other plastics in the oceans, which are bioaccumulating. So we have a lot of issues that we have to deal with. And this is where the idea of biotechnology and bioengineering can address some of these problems. And so my research has focused on bioengineering for biofuels and bioproducts. The first 10 years of my work here at the University of Manitoba, when I came in 2006, over the first 10 years we were looking at biofuels. Of course, then the price of oil dropped. Nobody really cares about biofuels anymore. So there's no money for research for biofuels, so we focused on the bioproducts. Well, what we do basically is industrial microbiology, using microorganisms to convert waste into products of value. Right? When we are drowning in waste, we have abundant waste. It should be seen as a resource that we can then recycle, reuse, and make into products of value. And these include biofuels, biopolymers, antioxidants, and even applying microbes to get rid of some problems to sort of in terms of bioremediation. So I'll talk a bit about that later too. So the work that we do is multidisciplinary. So students in my lab are from undergraduates in chemistry, biochemistry, microbiology, biotechnology, with genome sciences as a kind of fundamental platform to understand the microorganisms that we're working with. What's the genome, what genes are in the genome, what's expressed at the transcription level, at the translation level, and what products, what pathways are utilized to make what products. And then we have the biosystems and bioprocess engineering to how do we scale those up? How do we apply those to practical problems? So it's very, very exciting to me and been very, very fruitful. So in the 13 years...
Could you hit the PowerPoint slideshow?
Oh, okay. Sorry about that. I got carried away. I thought I had already done that. There we go. So sorry. Thank you. All right, so should I start over again? No. No. So from 2009 to 2014, I was the co-lead on a large-scale applied research project funded by Genome Canada with Richard Sparling in microbiology called Microbial Genomics for Biofuels and Bioproducts. This was a $10 million program, international, with researchers across Canada, US, and New Zealand. Then I got involved in BiofuelNet, which was a network center of excellence program with 180 partners, academics, industry, government, and I led the Prairie platform because it was divided into regions, and we were looking at ways of using cellulose to make ethanol and hydrogen, which led into the hydrogen, where kind of dovetailed with the Hydrogen Canada network, which was a $5 million project over 5 years, $1 million a year, with 26 researchers, 13 universities, and 4 NRC institutes. And I led the hydrogen production and purification team. So all of these things fit together. They're all kind of interlocking, and they're all basically, we're using the same approach where we were using genome sciences, bioinformatics, genomics, proteomics, transcriptomics, metabolomics, to understand and identify and develop microbial metabolism for biofuel and bioproduct production, looking at novel bioprocess-ready consortia, like looking not just at individual microorganisms, but communities or small communities or consortia of microorganisms to enhance conversion of lignocellulosic biomass to fuels, ethanol, hydrogen, and high-value co-products. This is the key about biofuels, that the biofuel itself is the lowest value product you can make, and economically it really can't compete with petroleum. So you have to make higher value co-products that make it economically viable. So for example, Husky Energy makes bioethanol from wheat and from corn, or corn-wheat mixes. But the real value stream comes from the dried distillers grains, not from the ethanol, right? So it's really a dried distillers grain industry that makes a side product of ethanol rather than a biofuel, right? So this is part of the problem with developing a bioeconomy, that it can't compete with the petroleum industry, and you have to have these co-products, and then it's a whole other story, which a whole other lecture that I won't go into today. Anyway, so the Genome Canada thing was incredibly successful. We had 4 invention disclosures, 3 patent submissions, 5 industrial partners, 2 spin-off companies, 8 novel bacterial strains deposited with repositories, 12 genome sequence submissions and announcements, 4 bioinformatic databases and tools developed. One of them a number of them developed by Brian Frostinsky here in agriculture. He was really a great collaborator. A bunch of media announcements, 77 publications, and 33 book chapters. So it was really successful. But because the price of oil dropped and nobody cares about biofuels anymore, it kind of ended. But not completely, because one of the side projects or sub-projects within that large-scale genomics project was isolation and characterization of bacteria capable of synthesizing biodegradable polymers, these polyhydroxyl alkonates. And so my research associate went to a hog barn and collected waste and then basically isolated bacteria that could synthesize these polymers. And we can visualize them by fluorescence. Oh, hello. Ooh, a bird. They can synthesize these polymers as internal granules, and they can be stained with a Nile red or Nile blue, and you get these little balls, fluorescent balls inside the cells, and that's how we isolated them. And so why is this important? Well, if we look at the world in which we live, we're surrounded by plastic. Everything we have, the tabletop, the computer things, everything is plastic. Even our clothes basically are made from synthetic polymers. So we can divide polymers into 4 kind of groups, right? Those made from petroleum, petrochemical, which either are non-degradable or degradable. So conventional plastics, polyethylene, polypropylene, polyethylene terephthalate, polystyrene, polyurethane. These are all conventional plastics made from petrochemicals, and they're non-degradable, or I shouldn't say non-degradable, but they're incredibly recalcitrant to biodegradation, and they take many, many, many years, and we don't really understand why, right? So that's another story we'll come to. And then on the other hand, there are some petrochemicals that are degradable, so PBS, PES, PCl. So they're petrochemicals derived that are biodegradable. And then you have renewable resources, polymers made from renewable resource, that are bio-based, but they're non-degradable. So bio-PE, polyethylene made from biosource, or polypropylene. They're made from a renewable resource, from agricultural products, but they're not biodegradable. So we don't really care about those. We don't want those. What we want are renewable polymers made from renewable resources that are biodegradable. So polylactic acid, polyhydroxyalkoxonates, starch-based polymers. And this is the quadrant where we would like to have polymers that have physical and thermal properties that are similar enough to petroplastics so that we can displace the petroplastics and replace them with biodegradable ones. So that's sort of the holy grail of our polymer research. So let's look into this a little bit more closely. So most petropolymers consist of only one type of subunit, right? So very simple, based on petrochemistry and very low-cost feedstocks. And you can make polyethylene, polypropylene, acrylic, polyamide, polyvinyl chloride, polycarbonate, etc. Now, because they have such a simple structure and it's always consistent, they have the same physical and thermal properties all the time, right? So the same polymer always has the same physical and thermal properties. They can be modified by the addition of chlorine, or bromide, or aromatic rings and things like that to change the melting point a little bit or change the elasticity a little bit. But basically, they're more or less the same. The advantage of PHAs, the polyhydroxyalkoxonates, is that renewable, they're biodegradable, and they're biocompatible. So anybody ever had oral surgery? I've had oral surgery, right? So they put sutures in your mouth, and then eventually they disappear. Well, those sutures are made from polyhydroxyalkoxonates, from very short chain PHAs, because these PHAs can either be made of short chain length subunits that have 4 or 5 carbons in them, or medium chain length PHAs which have 6 to 14 carbon subunits in it. And you get these kind of chains that either this is a short chain one or a medium chain long one with different chain length side chains. And the composition of these polymers determines their thermal and physical characteristics. And you can see that if you had a polymer that is 100% 4-carbon subunit consistently, that it would stack on itself very, very easily, right? It would become very crystalline. So you can get very high tensile strengths, but not very elastic. Whereas the medium chain length ones have variable chain lengths that interfere with stacking, with crystallization. So depending on the length and the proportion of the longer side chains, they're not even crystallizing, not even a solid at room temperature. They're a liquid that's sort of sticky or tacky, right? So we can vary the physical and thermal properties depending on the side chain composition. So in my lab, we use different microorganisms and different carbon substrates to grow them, and then they produce polymers with different compositions. So for example, if we use this bacteria, Cuprovitus necator, and grow it on glycerol, or waste glycerol from biodiesel, we get 100% C4 chain. If we mix fructose and valerate, we get a mixture of 75 and 25. If we then use, we can genetically engineer to change the enzyme that does the polymerization, we can make a polymer that's almost 100% or over 90% C5, a little bit of C4. And as we change the carbon chain, the subunit composition, we change the physical and thermal properties. And if we then do sort of medium chain or long chain fatty acids from oils, for example, we get more and more side chains with different carbon chain length, and we even get unsaturated side chains with 1 or 2 or 3 carbon-carbon double bonds, which actually is very interesting because then you can play with the chemistry and you can oxidize them so they cross-link to other things, which I'll come to in a minute. So basically the subunit composition determines the physical and thermal properties, and we can tailor the polymer composition to particular applications. So if you see this is polyhydroxyl butyrate, a polymer that's 100% 4-carbon subunit. It's very crunchy and very brittle, but it can be extruded into threads, which is what they use in the sutures. You can make bottles out of it, but they tend to break if you hit them, if you drop them on the ground. If we have a polymer, a medium chain length polymer made from octanoic acid, it's sort of 90% C8 subunit, and we get a nice transparent thin film, which is interesting. Or if we make it from glycerol, it's almost rubbery in composition, or rubbery in properties. So we can make lots of different things. And then we measure the physical and mechanical properties, so like tensile strength, elongation to break, elastic modulus, and the molecular mass, or the size of the polymer for polymers made from these different substrates. And we see that the polymers with the short chain length composition have very high tensile strength, say compared to low density polyethylene, 10.7 tensile strength versus 16. There we go. But they're not very elastic, where polyethylene has is very elastic, whereas PHB, which is 100% C4, is very brittle. It doesn't stretch at all. If we add a little bit of valerate to it, it becomes a little bit more elastic, but not as much as this. And the molecular weight decreases as the chain length, the subunit chain length increases. And we can also measure the thermal properties. Again, the short chain length PHAs have very high melting temperatures, 178, 169, 94, compared to, say, low density polyethylene, 108. But as the chain length increases, the melting temperature decreases, and the heat of enthalpy basically is when you melt the polymer, you melt the plastic material and solidify again, and then you remelt it, and it's a measure of the energy it takes to remelt it. And if it's like, if you take an ice cube and you melt it and then you refreeze it again, it becomes a solid again. It's got the same properties. But for some of these polymers, if you melt it and let it re-solidify, it doesn't come back to the same level of crystallinity and solidity that it had before you first melted it. So the second melt takes less energy, for example. Okay, so for example, 77 versus 8, right? Because it's not really crystallize, or it may take a longer time to crystallize. So these are some of the properties that we have to think about when we're trying to make polymers that can substitute for petroleum polymers, for example. Okay, so we talk about tailoring the PHAs for different applications. So how do we do that? Well, we do that by basically molecular biology and genetic engineering. And the key point here is that in the world of bacteria that make these kinds of polymers, there are actually 4 different types. The key enzyme in synthesizing these polymers is something called PHA synthase, encoded by the gene PHAC. So there are 3 kinds of PHA synthases that result in synthesis of short chain length PHAs. And only the pseudomonads, produce the medium chain length ones. And the key gene here is this PHAC, although there are actually two PHACs in the Pseudomonas, PHAC1 and C2. And in 3 and 4, there are actually two genes that encode different polypeptides, and so the PHA polymerase is a dimer, a heterodimer of two different proteins made by these two different genes. But we're not going to talk about that. We're going to talk about this. So in collaboration with Trevor Charles at the University of Waterloo, we did an interesting experiments where he had made a metagenomic library from soil, and from that had isolated different PHA synthase genes from the environment. We didn't know what they were. He has about, I don't know, 24 clones of these PHA synthase genes, PHAC genes, and he gave us one called clone 16, and we took our Pseudomonas putida LS46 and we deleted the operon that encodes the endogenous genes. So there was the PHAC1 and C2, and in between those there's something called PHAZ, which is the depolymerase, which I'll get to at the end of this. And so we deleted this and we substituted this clone 16. And if we look at the predicted three-dimensional protein structure of the endogenous PHAC synthase, it looks like this. And the predicted three-dimensional structure of the cloned gene looks like this. So you can see immediately from the three-dimensional structure that they look different. And when we grew the bacteria on the substrates, glucose, hexanoic acid, octanoic acid, nonoic acid, decanoic, and free fatty acids from biodiesel production, using the wild type, we get polymers with different compositions, right? So if you grow it on glucose, we get almost 70% C10 and 20% C8 subunits, a little bit of and C12. If we go hexanoic acid, it's like 80% C6, 20% C8. Octanoic acid is over 90% C8 and a little bit of C6 and C10, et cetera, et cetera. But when the recombinant that expresses this exogenous polymerase gene, growing it on the same substrates, we get fairly consistent short chain polymers with a little bit of medium chain polymer, right? So on glucose, instead of getting 70%, 20% C10, C8, we get 50% C6, 32% C4, 15% C8. If we go on hexanoic acid, it's 70%, 70-30, et cetera, et cetera. So these polymers will have very different thermal and physical properties than these polymers. So, and the only thing we changed was one gene, right? And the interesting thing is that now that this is sort of like a mixture of short chain and medium chain PHAs with a large portion of the C4 and C6 and a little bit of C10 in these ones, but not here. So these might have physical and thermal properties that are more interesting in terms of competing with petroplastics, with polyethylene or polypropylene. Well, we're still working on these. We still have to determine their physical and thermal properties to see how they behave. Anyway, but what can we do with them? Well, in collaboration with Song Liu in Biosystems Engineering, we did this electrospinning. We took the PHAs that we could make in the lab. So we had a PHBV, or polyhydroxyl butyrate valerate, with 25% valerate, 75% butyrate, or a commercial PHBV, which with 12% valerate and 82% butyrate. And he used this apparatus to basically where the polymer is in a syringe and it's drawn to a surface using a high voltage differential, a DC voltage, and creates these sort of nanofibers. Okay? So this is made from the commercial PHA. This is made from our PHA in the lab. And another interesting thing is that he could make these coaxial fibers where you have an outer coating with the PHA and an inner coating with a different polymer that contains a bioactive molecule. And the idea is that you could make these nanostructured materials that contain an antibiotic inside, and that if you were using it for wound dressing, for example, if your wound was infected, then the bacteria would secrete an enzyme that breaks down the PHA and creates holes in the core, in the shell, releasing the bioactive molecule to limit the growth of the bacteria. So these sort of Very interesting idea. And this is an electron micrograph of a cross-section of those core shell fibers where you can see the holes with these hollow tubes inside where you would have this bioactive molecule. And that work is still ongoing kind of thing. Another application is making biocomposite materials, and this is in collaboration with Dr. Ning Yan at the University of Toronto, Department of Forestry. And we gave her some of our PHA polymers that were made from canola oil or from flax oil. And the canola oil has about 5% of unsaturated side chains, and the flax oil, canola has about 5% unsaturated. Polymer made from flax oil, long chain fatty acids, has about 50% unsaturated side chains. And so she mixed it with about 40% cellulose and some epoxy, and we got a material that was like fiberglass. So if we look at the physical properties, the tensile strength, the specific strength, the toughness, if you compare the paper and epoxy with the PHA, it does better than just epoxy with paper. So adding a little bit of PHA, like 3 to 5%, increases the strength of that material. And if we compare it to fiberglass, say 25% weight of glass fibers with epoxy resin, the specific tensile strength is better than fiberglass. So we made a biocomposite material that's better than fiberglass, which was pretty exciting. Okay, so another issue about these things, if we're going to make all these different kind of polymers and we're concerned about the environment, we don't want to make strange and new polymers that are not going to degrade in the environment. So are they biodegradable? So now these bacteria make these PHA polymers as granules in the cell, basically as energy storage molecules. So when they're faced with an environment where they're carbon limited, they can mobilize these polymers and break them down and then metabolize them through beta oxidation to produce energy, right? So the enzyme that carries out that degradation is this PHAZ, or the depolymerase. And it's one of the many proteins. These granules consist of the polymers surrounded by a membrane, and there's a bunch of proteins embedded in the surface. Amongst these, the PHA synthase that synthesizes the polymer, and the PHAZ depolymerase that depolymerizes it when it wants to use it. So we developed a plate assay where we made colloidal plates of polymers made from different sources, from hexanoic, octanoic, decanoic, or long chain fatty acids. And we screened for a bunch of bacteria to see if they could grow on those polymers. So what we're looking for are these zones of clearance, which indicate, this is the best example here, that indicate that the bacteria are secreting some kind of enzyme that's degrading the polymer outside of it. So we found a few bacteria that did that, and then we cloned the PHAZ gene, the one that depolymerizes the polymer inside the cell when it wants to use the polymer. We cloned that into E. coli and expressed it, and then did the same kind of plate assay looking for zones of clearance. So now we have expressed this PHAZ protein, the depolymerase protein, and it's being secreted into the environment, and it's now degrading the polymer around where the bacteria is. We also developed a colorimetric assay using paranitrophenol connected to these alkanoates, basically of different length. And when if there's an enzyme that cleaves the octanoic or the alkanoic tail from the paranitrophenol, you get a yellow color. So we can actually quantify the thing. So this is a substrate blank. This is the PHAZ crude extract, and this is the purified protein. And basically we found that we got very, very high activity when the paranitrophenol is attached to a 4-carbon alkanoate, a little less when it's attached to a 2 or 8, and much less when a 12. But interestingly, this enzyme could also break down the petroleum-derived polymer, the PES, polyethylene succinated polyepsilon caprolactone, the PCl. So this enzyme that breaks down PHA inside the cell can also break down petroleum polymer. the biodegradable petroleum polymer, which is pretty exciting. And if you look at the structure of the PHA, this is the subunit structure of PHA and compared to polyethylene succinate and to PCl, it's kind of similar in a sense. You got this carbonyl group on the first carbon, and then an R group, a variable chain length over here. But here you have, so similar. So the enzyme must recognize this structure here, this ester structure with the carbonyl. And it depending on the side chain length or the arrangement of atoms around that, it determines the effectiveness of that cleavage. So that's something else that we could look at. So the take-home message basically are that PHA polymers are biodegradable and biocompatible. The rate and extent of degradation may vary amongst bacteria and depolymerase enzymes because each of those bacteria that we looked at have different abilities on different lengths of molecules. This great flexibility offers potential for making a wide range of applications. And polymers with different physical and mechanical properties, thermal properties, can maybe used to manufacture novel materials for new applications, which maybe one day, someday could displace or hopefully replace petroleum-based polymers. Okay, so that's the end of that section. How much time do we have left?
Another
15? Perfect. Okay, great. All right. So changing gears a little bit, changing focus. I want to talk a little bit about bioremediation and environmental sustainability. Of course, we know that that petroleum basically runs the world, and there are major problems with petroleum spills everywhere, right? The tar sands or the oil sands in Alberta are a kind of disaster waiting to happen with the tailings, and the spills in the ocean, the Deep Horizon disaster in the Gulf. So we're getting lots of petroleum spilled into the environment. And what do you do about it? Well, I developed a collaboration with Professor Simona Di Gregorio at the Department of Biology in the University of Pisa. And she had a project that was financed by the Fondazione Pisa, or sort of a Pisa Foundation, from 2016 to 2018. And they had an industrial partner. And the idea, the objective was to develop an engineered biological process to convert contaminated sediments and soils into decontaminated matrices resembling productive soil. So basically taking soil or sediments that are contaminated with hydrocarbons and remediating them into productive soils. So that's a big task, right? So part of this was to looking at the humified and stable organic matter that is structurally and functionally safe. That means without phyto or cyto or genotoxicity. So how do you do that? Well, they developed a process called BioResNova, which is an enhanced and automated windrow composting or form of land farming, which transforms organic contaminants into non-toxic compounds with simultaneous humification of the organic fraction. So this is an on-site or ex situ. It can be done on-site, or in many cases when they're doing bioremediation, they have to transport the soil or the sediment somewhere else, which would be ex situ. But it's based on traditional land farming or windrow composting. But it's been upscaled and involves now online monitoring of CO2 production, humidity, temperature, and microbial community. Right? And this is the important part that I'll get to in a minute. So the idea is to take contaminated soils and you grow up fungi that can break down polyaromatic hydrocarbons or total petroleum hydrocarbons that are in the soil, and you mix the fungi with the soil, and then you put it into windrows. And then you have a containment. You put it into a building, so you have the windrows in a contained environment where you have all your monitoring equipment. And then they have this machine, which is called RoboNova. And the machine basically goes up and down the windrow and mixes, sort of aerates, but at the same time takes samples for GC analysis, HPLC analysis, and microbial diversity analysis. So it's pretty high-tech stuff. And basically, what the example they have is that in Pisa, in Italy, there's a canal called the Navicelli Channel. This was built back in the 16th, 17th century when Pisa was actually one of these city-states in Italy that was very, very powerful because they had their own navy and they traded all over the Mediterranean, and they fought wars with Florence and with Milan to kind of vie for power. Those days are long gone, but they had this channel that was actually designed by Leonardo da Vinci. And it's all there along this channel that they had petroleum refineries, which are now shut down, they're out of business, but a lot of petroleum products seeped into the channel and ultimately into the harbor, and they have to dredge the sediments and try to remediate them. So they dredged the sediments. In the sediments, there's like this microbial community is less than 1,000 per gram of the sediments. The total petroleum hydrocarbons are very concentration is 2,800 parts per million, very high. Very low humic acid and very low fulvic acid content. So they took this material and they submitted it to the to the Bioresnova thing, and what they found was that there's a correlation between degradation of the total petroleum hydrocarbon degradation and the microbial community. That over time, certain operational taxonomic units, or certain kinds of phyla of bacteria, became more prevalent as the TPH, as the petroleum hydrocarbon concentration decreased. So by day 18, we had very little contamination, and you saw the appearance of these Bacteroides bacteria and these actinobacteria here, right? They also found that as the process continued, right, as the TPH concentration was decreasing sort of by, very, very low by day 18, the humic acid and fulvic acid content was increasing. So even by day 10 and up by day 28, you still had an increase in humic acid content and slight increase in fulvic acid. And now fulvic acid and humic acid are the polyaromatic molecules in soil that give soil its fertility, right? So by decreasing the hydrocarbon, basically we're converting the enzymes secreted by the microorganisms are converting the polyaromatic hydrocarbons in the TPH into fulvic and humic acid-like molecules, which is pretty cool. And then finally, they looked at detoxification. So while in the first 10 days there was an increase in toxicity, there was thereafter there was a significant decrease in toxicity. So basically the hydrocarbons were depleted, the humic acid, fulvic acid increased, and the toxicity decreased correspondingly. And that all correlates with the appearance of these this particular microbes in the population. Now, in addition to that, they looked at the frequency and the presence of certain genes. So there are enzymes called lactases, which basically oxidize polyaromatic hydrocarbons or polyaromatic molecules. And so pre-treatment, you had certain classes of lactases, but a few of them. But post-treatment, they had a huge increase in the number and different types of lactases in the sediments. Sort of again, kind of reflecting the change in the microbial community through this decontamination process. So that was pretty cool. So the take-home message here is that this process, this Bioresnova process, can remediate and detoxify contaminated sediments and humify and stabilize the matrix to restore its functionality and its productivity. And so both fungi, so microremediation, and bacteria play a critical role. And it's, there sort of creates an opportunity that you can provide better or enhanced bioremediation technologies by treating hydrocarbons in soil and sediments and other places with this biological process and monitoring the process as a quality control, because as you can see the changes in the microbial community that are correlated with changes in the matrix, which is pretty cool. Okay, last story. So continuing with this bioremediation, environmental sustainability, and this issue of plastic. We have plastic everywhere. So we started a little project looking at, can microbes really degrade plastic? or polyethylene in this case, because polyethylene is the most abundant single-use plastic, right? Very recalcitrant to biodegradation. Some estimates are 500 to 1,000 years. But microbial degradation has been observed, and if it but we don't know how much or why it doesn't go to completion. So there are 4 steps to bioremediation or biodegradation of plastic, basically. So you'd have biodeterioration, deterioration of the plastic, fragmentation of the chains into shorter molecules, which then would be assimilated by metabolism into the bacteria, and ultimately, hopefully, mineralization, where you have all of that material, that substrate, converted into carbon, basically biomass in terms of bacteria, carbon dioxide, and water. But it seems to stop here. We don't know why. So there are many studies of microbial degradation on polyethylene, but the results are very, very highly variable in the literature. Some cases, months, takes months for different kinds of bags, and the weight loss, measured by weight loss only, maybe 20 to 30% or 2%, 9 to 20%. So very, very variable, not very convincing. So we thought we could do better. So I had a visiting student from Iran who was working on this project in Iran, and she had isolated bacteria from a landfill that was full of plastic. And she wrote to me and said she would like to come and work in my lab. And I said, well, I don't work on plastic. I work on biodegradable plastics. But she wanted to come, so I said, okay, let's try it. So she came. She brought some of her bacteria. And we had 4 well-characterized species in our lab that make PHAs, but are also ubiquitous soil bacteria, and they're known to be hydrocarbon-degrading bacteria. So we thought we would try it, because if we find something, we could correlate it with the genome, because that's what we do, right? So what we found, what Zara found, was that what she did was actually take the bacteria and cultivate them in pre-cultures with mineral oil. So mineral oil consists of alkanes, linear hydrocarbons that have single bonds between each carbon. So she grew it on hydrocarbon first, and then over time reduced the amount of hydrocarbon, increased the amount of polyethylene, and these are polyethylene powder, essentially. And so until it was growing on 100% polyethylene. And then we did the experiment. And what she found is after pre-culturing those bacteria with hydrocarbons, within 3 days, we got very high growth and very rapid breakdown of the polyethylene. Then they kind of tapered off by day 10, and then we don't know what happens out here. Some of them, they went down, and then they kind of started going up again. We don't know exactly what's going on. But this very, very rapid degradation was very evident. We found that if we look at cell mass production, we had this Cuprovitis necator, which synthesizes these short-chain PHAs, had very high highest biomass production compared to the others. The reduction of the polyethylene biomass, or polyethylene mass, these 3 bacteria, the Cuprovitis necator and 2 Pseudomonas putida species, including the one that we use to make PHA, had the highest, between 30 and 35% reduction in the mass of the polyethylene. And we are able to isolate hydrolysis products from the polyethylene. So carbon chains, linear carbon chains that were fragments left over after the bacteria ate. So these are different lengths of hydrocarbons, from 2, 3, 4, 5, 6, 7, 8 carbon chain lengths up to whatever. And this is the stuff that's left over, the stuff that's not eaten. All the other stuff that's 0 is the stuff that the bacteria assimilated and ate. So these different species have different food preferences, it seems like. They don't like this one, doesn't like these ones, this one doesn't like these ones, et cetera, the C. necator just doesn't like those 2. And our Pseudomonas putida eats everything except that one, right? So that's pretty cool. And so the take-home message for this is that there's a link between hydrocarbon degradation and plastic degradation, because after all, plastic are hydrocarbons that are cross-linked, right? Polyethylene is basically linear alkanes that are cross-linked. So it makes sense that the enzymes that break down hydrocarbons can also attack the polyethylene. Although this is a hypothesis that's still yet to be proven, we need to do proteomics and understand which enzymes are being secreted when the bacteria is growing on hydrocarbon, and are they the same enzymes when it's been converted to grow on polyethylene. So this is a story that's still continuing. And yeah, so that's pretty exciting stuff. All right, just to finish off, some new initiatives. So recently got a grant from Agriculture Canada, the Ag Science Bioproducts Cluster, to use our PHAs to engineer PHAs that have thermal and physical properties that are similar to polyethylene terephthalate and PE, and develop smart packaging materials with blends of different biodegradable polymers. So that funding is approved. I have another project that's just starting with International Institute of Sustainable Development at the experimental lakes. They're looking at hydrocarbon degradation of crude oil and bitumen spilled into pristine boreal forest lakes. So I have a new PhD student working on that, and that funding is approved. And in the process of putting together one of these new frontier transformation stream applications, basically looking for bio-based technologies for degradation of petroplastics. So can we take some of these ideas that we've been working on for polyethylene or the bioresnova and develop and find bacteria and look at the enzymes and try to understand the mechanisms of breakdown of polyethylene or other plastics and why it stops, doesn't it go to mineralization, what are the limitations. So this is a very large-scale international multidisciplinary multi-institutional research team, and the application is just in the process of starting. We just submitted our internal EOI, so who knows? We'll see what happens. Anyway, that's the story for today. Any questions? Oh, wow.
Give the present.
Wow, a present. Okay. Can I open it now? Okay, a notebook. Excellent, thank you. Yeah, that's true. I'll need to write them down.
That was fascinating. I really enjoyed that. So, is there a question or more questions? Yeah. One there.
For these bacteria that produce the plastics or the polymers, what do you need to feed them in terms of, I'm just looking at energy costs and what actually goes in and comes out of the system.
Right.
So
Well, yes. So there's the rub, right? Any bioprocess is going to cost more than any petrochemical-based process, and if we there's a disconnect. I think everybody in the world recognizes that we're facing this big problem with petroleum plastics. The petrochemical industry is continuing to produce them, right? The cost of feedstock is so low because of fracking of natural gas that it takes pennies to make polyethylene, polypropylene, all of these things. And biodegradable polymers, or PHAs, or PLA, or all of these things, are bio-based processes or renewable processes. They're going to cost way more. And so unless there's a disincentive to buy petroleum plastics and some kind of incentivization to buy renewable plastics, biodegradable plastics, it's going to be same old, right? In Europe, though, they're way ahead of North America. In many jurisdictions in Europe, they've actually banned single-use plastics, polyethylene in particular. So you can't get a polyethylene plastic bag in the grocery store. They're all PLA plastic bags now. And in Germany, they're actually penalizing not the distributor, but they're penalizing the producers of the plastic. There's a tax or a tariff, like a carbon tax, on the production of petroplastics. So these are the kinds of things that are going to happen to equalize the balance in the cost. And yes, of course, we try to use substrates that are waste streams, so waste glycerol from biodiesel or waste-free fatty acids, sugars from cellulose, right, which in itself costs more because you have to break down the cellulose to make the sugar. So there's going to be a lot of work to develop a commercially viable process using bioprocessing and bioproducts to make bioproducts. So a big question.
Well, I was just curious, in terms of seeing, like I'm trying to imagine what the commercialization and industrialization of this would actually look like.
Well, it would look like ethanol production, million liter fermentation tanks, yeah. Yeah, with downstream processing, right? It would be exactly that, because these are products that are made internal to the cell, so the cell has to grow. Then you have to dewater it, you have to concentrate it in a centrifuge, and then you have to break open the cell and precipitate the polymer away from the cell mass, et cetera, et cetera. So, yeah, yeah.
You mentioned cellulosic ethanol at the beginning.
Yeah.
Did you solve that? Is it?
No, nobody solved it, right? The problem, there were a lot of cellulosic ethanol plants that were opened in the States, and a big one in Italy, in northern Italy, that were based on straw, and they've all gone out of business because the price of oil is so low and the price of gasoline, they can't compete. Because there was at one point during, the Bush administration and the Obama administration, there was the renewable fuel standards, right, where they give subsidies to the ethanol companies, which were either big ag industries like Monsanto, because they're using corn in the States, or petrochemical companies like Husky in Canada. But all of those subsidies have now been eliminated, so they can't compete, right? I mean, Husky's still. The process is, oh yeah, processes is possible, sure. I mean, sure, these industrial microbiology, I mean, you make beer, that's industrial microbiology, right? You make ethanol, it's the same, it's the same thing, and a lot of bioproducts are being developed. There's some other companies that are producing xylitol and other sweeteners and things like that, but the bioproduct has to have a really high value in order to justify the cost of production, right? And has to be competitive with other things, right? So, yeah.
And my question, David, is about what these biodegradable polymers degrade into.
Yeah.
My semi-ignorant or largely ignorant understanding of biodegradable plastics is that with oxygen, they degrade into carbon dioxide partially. If we bury them and they ferment in the soil, they degrade into methane. These are carbon-based biopolymers. Wouldn't they do the same thing?
Well, the degradation is caused by microbial action. It's enzymes secreted by the bacteria that cleave the polymer into smaller fragments, and then the bacteria eat them. But as we've shown here, they don't eat all of it. They eat some of it, but not all of it. There's some residual long-chain fatty acids and things like that. But for PHA polymers, the subunits are 3-hydroxy fatty acids. So they're very similar to the fatty acids you find in oil, right? So the anaerobic side is if you're anaerobic, then you're going to get methanogenesis. Right? They're going to eat whatever the carbon source is. Methanogens are going to take that and they're going to make methane. So whether it's PHA polymers or residual oil or whatever, if it's an anaerobic environment, you're going to get methane production. So it's not exclusive to these polymers. But basically, in an aerobic environment, they will disappear, and it only takes, it takes about 3 to 6 months for a bottle that's made of PHA to completely mineralize, to disappear. And it produces biomass in terms of bacteria and carbon dioxide and water, right? Yeah, so there was a question.
Yeah, so I really enjoyed that talk. It was a little bit outside of my range at this kind of bit. I have one business question and 2 science.
Sure, yeah.
So my business question is, like, what is the business case for bioremediation? Because it clearly, like, does good, but, like, given that biofuels...
Right, who's going to pay for it? Who you're asked. Good question, because industry, the polluters, don't want to pay for it, right? There's so many examples of oil and gas companies that develop a site, and then when they're finished with it, they just leave it behind, and it's completely contaminated. And my biggest fear, and I think many people's biggest fear, that's what's going to happen in the oil sands too, right? You have tailing ponds that are humongous, and it's terrible. But they are forced by government policies to invest in cleaning up. And it's whether the government will enforce those to make sure that they do. But there are so many examples of gas stations or oil storage locations where tanks have leaked and they're contaminated the soil, and they just walk away, and if somebody wants to develop it, they have to remediate. They have to take that soil out and replace it with non-contaminated soil. Happens all the time here in Winnipeg even. So that's the first one. Yeah.
Okay, and then my two science questions are, the first one is that I noticed in your Pseudomonas, like clone 16, that the D polymerase was nestled in between those two, like PHA polymers.
Yeah, that's the normal condition. Yeah.
Have you considered deleting it to
Oh, yeah.
potentially, has that worked?
Well, yeah, you can delete. We've deleted the entire operon. We've deleted the polymerase 2 gene. We've eliminated the polymerase, the depolymerase and the polymerase 2 gene. It doesn't. That depolymerase is there for when the bacteria is living in the wild and finds itself in a condition where it doesn't have any carbon to eat. In the lab, we're feeding it carbon all the time, right? So it doesn't really need the depolymerase. So you can delete it without impunity if in a lab strain, because it won't grow unless you have carbon, right?
But it doesn't, like deleting it doesn't increase yield at all?
No. But we did find that the genes that control PHA production are controlled by quorum sensing. And in bacteria, like the Pseudomonas chloraphus produces phenazine and another antibiotic, and you delete those genes, then PHA production goes up because you're not diverting carbon and nitrogen and energy into antibiotic production. So.
And then my last science question is, I noticed in one of those last slides with the bioremediation and comparing the byproducts among the different strains in bacteria that you use.
Yeah.
Have you considered, because there was that one on the last
Yeah,
slide,
yeah.
Pseudomonas that just leaves the one,
Right.
and then the other ones seem to eat it. Have you considered like combining them together?
Well, yeah, so using consortia, that's definitely an approach that if we had more money, we would do. And hopefully, if we get this new frontiers transformation thing, that's exactly what we're going to do. And not only with different bacteria, but with fungi as well. Sort of kind of making these sort of microbial communities that are specialized in degrading recalcitrant compounds. That's the way to go, for sure. Yeah. Good questions. Yeah.
Krista Meyer and I were just talking about how excited we were about the idea of being able to remediate soils rather than sending them to the landfill.
Mm-hmm.
So is the idea that you can remediate these soils in these windrows and then take them back to the site?
Well, presumably, based on the data that they showed, that the toxicity and the hydrocarbon contamination drop below kind of the standards that are required, and you increase the humification, and you've got productive soil. So nobody's really tried this. In North America, this doesn't exist in North America. This is in Italy, and now it's sort of catching on well, it was a Pisa thing, and now it's catching on in Italy because a lot of government pressure to remediate the harbors and the dredged sediments, but it can be applied to land. I'm a co-supervisor, advisor on a student from Pisa was in my lab for a year and a half, and they're looking at soils from an abandoned oil refinery, and so they're remediating the soil using the same kind of technology. So it could be done with soil. Or if you had soil, agricultural soils, I don't know. I mean, that's a long road, I guess, but you'd have to demonstrate that it works and that it's safe, and then maybe that becomes a standard, and that would be wonderful. But it's just the beginning.
Is there one more burning question?
Yeah.
I have a question about these genetically engineered bacteria and what the regulatory requirements would be to actually release them into the
In Canada, I don't think there is any regulations in terms, if it's non-food, it's not a problem. But in these cases, for this Bioresnova thing, I don't think you'd be releasing it. It's still going to be in a contained building kind of thing, right? Or have some kind of covering so that it doesn't get blown downstream to somebody else's farm or something like that. But again, these are long-term issues that we have to demonstrate that the technology works consistently, and it has to be transferred from Europe to Canada and accepted, and so who knows? But I don't think in terms of GMOs, use of recombinant bacteria in the environment, no, I don't think there's there's no restrictions to that at this time, as long as it's not food. Right. Yeah.
Well, thank you very much.
Yep. You're welcome. Thank you. Thank you for coming. Great.
So I'm work on pesticides, and when we measure CO2 from the chemicals, we all use radio-labeled stuff.
Uh-huh.
How do you measure CO2 directly?
Well, I don't. I mean, the Italian people do.
Yeah.
I think they use
Yeah, I see. Okay, and do you do concentrations of hydrocarbons in your lab, like GC, MS, MS?
Yes,
or
yes,
else?
yeah. Well, we have GC. We don't have GCMS, but we have access to that in the chemistry facility or through Greg Toley. He's got a hydrocarbon lab, right? So if you're measuring hydrocarbons,
Yeah,
then
you go
there are-
through his lab.
Yeah,
Okay, so you're more the genomics guy.
yeah, we're the bacterial side.
Yeah, I was just wondering
Yep.
about other compounds and stuff. But yeah, it was great. I really enjoyed it.
Well, thank